For over a century, it has been taught as scientific fact, a foundational principle repeated in every medical textbook, every biology classroom, every museum exhibit on human disease. Tuberculosis, the white plague that killed more humans than any other disease in recorded history, came from our livestock, specifically from cattle. When humans domesticated cows around 10,000 years ago during the agricultural revolution, we caught a deadly pathogen that would go on to claim over 1 billion lives. It seemed logical, obvious even. After all, cattle get tuberculosis. Humans live close to cattle. Diseases jump from animals to humans all the time. For generations, scientists accepted this narrative without question. But in 2014, a team of geneticists led by Johannes Krauss and Kirsten Boss at the Max Planck Institute for Evolutionary Anthropology made a discovery so shocking it would overturn everything we thought we knew. By sequencing tuberculosis DNA from ancient human skeletons and comparing it to modern strains, they built a genetic family tree that told an impossible story. We didn't catch tuberculosis from cows, cows caught it from us. And the molecular clock suggests it happened not 10,000 years ago, but 70,000 years ago. The story does not begin in the dairy farms of Mesopotamia or the cattle markets of ancient Egypt. It begins in the laboratories of evolutionary biologists who were trying to answer a question that had puzzled scientists for decades. If tuberculosis came from cattle, why does the human version of the disease look so different from the bovine version? Why do the bacterial genomes have completely different structures? And most troublingly, why do ancient human skeletons show evidence of TB from thousands of years before large-scale cattle domestication even existed? These were not minor inconsistencies, they were fundamental contradictions that the conventional theory could not explain. But without the tools to read genetic code directly from ancient bones, scientists could only speculate. That all changed with the advent of ancient genomics, the ability to extract and sequence DNA from archaeological remains thousands of years old. The Max Planck Institute team, working alongside researchers from institutions across Europe and Africa, decided to construct the most comprehensive genetic family tree of tuberculosis ever attempted. In the early 2000s, researchers began collecting samples of Mycobacterium tuberculosis, the bacterium that causes TB, from every corner of the globe. They sampled modern strains from patients in hospitals across 6 continents. They extracted ancient DNA from the teeth and bones of people who died thousands of years ago, including victims from Neolithic settlements, medieval plague burials, and pre-Columbian American sites. They analysed tuberculosis bacteria from cattle, goats, seals, sea lions, elephants, and dozens of other infected species. By 2010, they had assembled over 1,300 samples spanning thousands of years and representing every major lineage of the disease. The task was monumental. Extract the bacterial DNA from degraded bone tissue. Sequence the entire genome, millions of genetic letters long. Compare these sequences across all samples. Build a phylogenetic tree, a family tree that shows which strains evolved from which ancestors and in what chronological order. This tree would reveal the evolutionary history of tuberculosis. It would show where the disease originated, how it spread, and most importantly, whether it jumped from animals to humans or the other way around. The first results came in 2012, but they were preliminary, based on partial genomes. Even so, they hinted at something unexpected. The human strains of TB appeared older, more ancestral than the animal strains. But the data was incomplete. The team needed full genomes from ancient human remains to be certain. They turned to archaeological collections, searching for skeletons with visible signs of tuberculosis infection. The disease leaves characteristic marks on bones, particularly the spine and ribs. A condition called Pott's disease causes vertebrae to collapse and fuse. These skeletal signatures allowed researchers to identify likely TB victims from the distant past. One crucial site was Atlit-Yam, a submerged Neolithic village off the coast of Israel. There, archaeologists had discovered skeletal remains dating to 9,000 years ago, showing clear signs of TB infection. In 2014, the breakthrough came. The team published their landmark study in Nature Genetics, led by Inaki Comas and colleagues. They had successfully sequenced complete tuberculosis genomes from multiple ancient human remains, including those 9,000-year-old skeletons from the Eastern Mediterranean. These were Neolithic people, early farmers, but their TB infection dated to before large-scale cattle domestication had fully spread to that region. Even more stunning, when the geneticists placed these ancient strains on their phylogenetic tree alongside modern human and animal strains, they found something extraordinary. The ancient human TB sat at the root of the tree. It was the ancestor. All other strains, including those from cattle, goats, and other animals, branched off from the human lineage. The tree was unambiguous. Mycobacterium tuberculosis, the human form, came first. Mycobacterium bovis, the cattle form, came later, as a descendant. But the evidence didn't stop there. The team analysed the genetic structure of different TB strains in detail, looking for what geneticists call regions of deletion, or RD markers. These are chunks of DNA that have been lost during evolution. They found that human TB has a more complex, complete genome. Cattle TB, by contrast, has a simplified genome with multiple regions of deletion. It's missing entire genetic sequences that human TB possesses. In evolutionary biology, this pattern is a smoking gun. Complex genomes don't spontaneously gain new genetic material to become simpler ones. Instead, simple genomes are created when complex ancestors lose unnecessary genes while adapting to new hosts. The regions of deletion in bovine TB proved it was a streamlined descendant, optimized for cattle physiology by shedding genetic baggage it no longer needed. The conclusion was inescapable. Human TB is the ancestor with the original complex genome. Cattle TB is the descendant with a reduced specialized genome. The disease evolved in humans first, then jumped to cattle thousands of years later, losing genetic material in the process. The implications were staggering. Every textbook was wrong. Every museum exhibit would need to be revised. The foundational narrative of tuberculosis as a disease of domestication had just been turned completely upside down. But if humans didn't catch TB from cattle, where did it come from originally, and when? The team turned to a technique called molecular clock analysis. This method doesn't require finding 70,000-year-old DNA, which doesn't exist because DNA degrades over millennia. Instead, scientists measure the rate at which genetic mutations accumulate in TB bacteria over known time periods. By comparing modern strains to ancient strains from dated archaeological sites, they can calculate how fast the bacterial genome changes. Then, by measuring the total genetic distance between all known lineages, they can work backwards to estimate when they all shared a common ancestor. Genetic diversity patterns suggested that TB originated in Africa, spreading with early human populations as they migrated out of the continent and across the globe. This means hunter-gatherers living in small nomadic bands long before the invention of agriculture were already infected. The disease was endemic, a constant background presence in human communities. Cave-dwelling populations were particularly vulnerable. Living in enclosed spaces with poor ventilation created ideal conditions for respiratory transmission. TB spreads through airborne droplets when an infected person coughs. Sharing caves meant sharing breath, and sharing breath meant sharing tuberculosis. Archaeological evidence from multiple continents supports this. Skeletal remains from pre-agricultural human populations show signs of TB infection. Ancient DNA extracted from these bones confirms the presence of Mycobacterium tuberculosis. The disease was global before agriculture even began. Hunter-gatherer bands were small, typically 20 to 50 people, and highly mobile. Infected individuals were naturally isolated by the group's movement patterns. Transmission rates stayed manageable. But agriculture changed everything. Permanent settlements brought hundreds or thousands of people together in close quarters. Enclosed dwellings provided better conditions for airborne transmission than outdoor camps. Malnutrition from grain-heavy diets weakened immune systems. Population density skyrocketed. And here's the critical part. When humans began domesticating cattle, goats, and other livestock around 10,000 years ago, they started living in extremely close proximity to these animals. Families shared sleeping spaces with their herds. Farmers worked alongside cattle from dawn to dusk, breathing the same air in confined barns and homes. This created the perfect conditions for reverse zoonotic spillover, the transmission of disease from humans to animals. Humans with active TB infections, coughing and expelling billions of infectious droplets, were in constant contact with their livestock. The cattle inhaled the bacteria, and over generations, tuberculosis adapted to its new bovine hosts. Mutations that helped it survive in cow lungs were selected for. Genes unnecessary for bovine infection were lost, creating those regions of deletion. The result was Mycobacterium bovis, a distinct strain optimized for cattle physiology. In fact, TB proved so adaptable that it even reached the Americas long before European contact. Ancient DNA from pre-Columbian human remains shows TB infection, but the strains are most closely related to seal and sea lion TB from the Pacific. The current hypothesis is that infected pinnipeds, marine mammals that haul out on coastlines, transmitted TB to indigenous coastal populations in South America thousands of years ago. The bacterium didn't jump from cows to humans. It jumped from humans to cows, from humans to seals, from seals back to humans. Tuberculosis is an opportunistic colonizer, adapting to whatever warm-blooded host it encounters. But then, in a cruel twist of evolutionary irony, humans began catching tuberculosis back from their cattle. This happened through a different route, raw milk. Bovine TB concentrates in cattle mammary glands and udders. When humans drank unpasteurized milk from infected cows, they contracted Mycobacterium bovis. This form of TB typically caused extrapulmonary disease, affecting bones, lymph nodes, and internal organs rather than the lungs. For thousands of years, humans were caught in a cycle of our own making. We gave TB to our livestock, then we caught it back through our food supply. It wasn't until 1864, when Louis Pasteur invented pasteurization, that this transmission route was finally broken in industrialized nations. Even today, in regions where raw milk consumption is common, bovine TB remains a significant public health threat. The revelation that humans are the original reservoir for tuberculosis has profound implications. It explains why TB is so uniquely adapted to human physiology. The bacterium has had 70,000 years to co-evolve with our immune systems, learning to evade our defenses, hide inside our macrophage cells, and persist in our bodies for decades in a dormant state. This is why TB is so difficult to treat. Standard antibiotic therapy requires 6 to 9 months of multiple drugs taken daily, far longer than treatment for most other bacterial infections. It's why TB has become so adept at evolving drug resistance. Multi-drug resistant TB, extensively drug-resistant TB, and even totally drug-resistant strains have emerged in recent decades. The bacterium has 70 millennia of experience surviving human immune responses. Modern antibiotics, invented less than a century ago, are just the latest challenge, and TB is adapting in real time. Today, tuberculosis kills approximately 1.5 million people every year. It is the leading bacterial infectious disease killer globally, surpassing even HIV and malaria in annual mortality in some regions. Despite a century of medical advances, despite the development of antibiotics and vaccines, TB remains one of humanity's oldest and deadliest companions. The reason is now clear. This is not a disease we caught from animals during the agricultural revolution. This is a disease that has been with us since the beginning, woven into the very fabric of human existence, traveling with us out of Africa, spreading to every continent, adapting to every environment we've inhabited. It is, in the most literal sense, a human disease. This discovery transforms our understanding of human history. Tuberculosis shaped our evolution. Immune system genes that help resist TB infection, as variations in the NRAMP1 and TLR genes, have been under positive selection pressure for thousands of generations. Populations with long histories of TB exposure show higher frequencies of protective genetic variants. The disease influenced population movements, settlement patterns, and even cultural practices around health and contagion. But perhaps most importantly, it reveals a humbling truth. We are not just victims of tuberculosis, we are its original home, its evolutionary cradle. Every animal that carries the disease today, from cattle to elephants to seals, caught it from us or from another animal we infected. We are the source. The phylogenetic tree, built from millions of base pairs sequenced from bones and living patients across the globe, tells an unambiguous story. Humanity and tuberculosis have been locked together for 70,000 years, and after all that time, this ancient enemy knows us better than almost any other pathogen on Earth. If you found this revelation about tuberculosis' true origin fascinating, hit subscribe to explore more stories where DNA evidence rewrites what we thought we knew about human history. Until next time,